Hi everyone, welcome to lab exercise 3.3 where we will be talking about gene expression or the flow of genetic information from DNA to protein synthesis. Today we're going to be extracting some DNA, but first let's talk about protein synthesis. This first page, including the QR code, is a review of transcription and translation. Remember, when we transcribe something, we're going to remain in the same language. So we're going from a nucleic acid DNA language to a nucleic acid RNA language. mRNA or messenger RNA is kind of like the shorthand of DNA. Also remember that adenine has 2 rings and always binds with thymine. Guanine has 2 rings and always binds to cytosine. This helps prevent making mistakes because, as you see, adenine and guanine are much larger than thymine and cytosine, so it's unlikely that they will be mismatched. Also, notice that adenine-thymine has 2 hydrogen bonds, where thymine and cytosine have 3 hydrogen bonds. So it's very unlikely that adenine will accidentally match with cytosine or that guanine will accidentally match with thymine. Also, remember that in RNA, thymine is replaced with uracil. Now, transcription happens inside the nucleus, whereas translation happens outside the nucleus, usually in the endoplasmic reticulum or the cytoplasm. Really, it's going to happen anywhere that there's a ribosome. So as we follow the flow of genetic information, we're going to start with DNA. We're going to transcribe into RNA. We're going to modify the RNA so that it's allowed to leave the nucleus, and then we're going to translate it using a ribosome into amino acids or a polypeptide, which eventually could become a protein. If you need a review of this process, you can scan this QR code and follow the instructions and actually transcribe your own DNA. But what we're going to be doing is we're going to transcribe and translate this strand of DNA. Remember, we always start out at the 5' end and end at the 3' end. On this second page, I have already copied the coding strand. Now, this is the coding strand, and this would be the template strand. So RNA polymerase is actually going to attach on the template strand, but it's going to make a complementary copy, which means it's most going to represent the coding strand. Let me show you what that means. When we are going through the process of transcription and translation, the first thing we're going to do is look at the template strand. Remember, we're always going from the 5' end to the 3' end. So if we're reading the coding strand this direction, the template strand will be anti-oriented, but it will be an exact complement of the coding strand. So we're going to start here. What matches A? T, because we're talking about DNA. T, A. T, G, T. We're going to find the complements here, C, G, C. If you're wondering why I'm complementing G with C, well, you can look back at the first page. C is always complement to G, A is always complement to T. So let me continue. C, A, T, T, C, C, G, C, T, T, G, C, G, G, C, T, G, A, T, C, C, G, T, C, A, T. Now, I'm going to act as if I am RNA polymerase. RNA polymerase only moves in the 5' to 3' direction, so I'm going to build in this direction. Now, I am transcribing the template DNA into RNA. It's not quite messenger RNA yet, it's pre-mRNA. and it's going to be complementary to the template strand, which means it's most likely going to resemble the coding strand. So I'm going to find the complement to the template strand, which is AUG. I can kind of check my work by looking up here, and it does appear to be very similar to the coding strand. Now we already know AUG is very, very important. It is the start codon. So we're going to pay attention to that. Now the complement here, I made a mutation, a mistake, ACG. Doesn't quite look like a C. Make sure that we know that's a C. GAU, CAG, CCG, CAA, GCG, GAA, UUG. Now notice, normally the complement would be thymine, but since we're talking about RNA, it's going to be uracil. GCG, ACA, and UAA. Now I'm done with pre-mRNA. I need to modify my RNA so that it can leave the nucleus. One thing that I'm going to do when I modify the DNA, I'm sorry, the RNA, is add a 5' methylguanosine cap. So on the 5' end, I'm going to add a G. On the 3' end, I'm going to add a poly A tail. So over here on the 3' end, I'm going to add a poly A tail. I wrote this in the wrong place. G. Now the other thing is that I need to remove the introns. These are labeled as introns, and they are not part of the final product. That doesn't mean introns aren't important though. Introns can be very, very helpful for other reasons. They're just not used in transcription. So when I copy down my mature RNA, I'm just going to copy the exons. Only the exons remain. I'm not going to pull down the introns. Now, these are going to move over here because I'm cutting out this section. Now, notice that my mature mRNA that's now allowed to leave the nucleus is much shorter than my original strand because I removed those introns. The next thing I'm going to do is translate. So far, I've only stayed in the nucleic acid language. DNA and RNA are both nucleic acid language. Now I'm going to switch to amino acid language to create a polypeptide or a protein, and that's going to be translation. Now this is going to happen anywhere there are ribosomes, mainly in the endoplasmic reticulum. What I'm going to do is I'm going to use this chart down here to find out which amino acid matches the codon. So the mature mRNA is our codon, and we are going to find the matching amino acid that's going to be pulled in by that transfer RNA or the tRNA. So I'm going to start with A, U, G, which is methionine, which is really important because it's the start codon. Methionine. Next, A, C, G. C, C, G. Now, if there were accidentally a mutation in this area, it probably wouldn't make a difference because all of these are redundant. As long as the first two are the same, when we're talking about proline, it doesn't make a difference, but this is proline. CAA, GCG, again, all of these are redundant, all of them are alanine. This GCG again, ACA, UAA. Now, this does not have an amino acid, which means it's the stop codon. It actually codes for a termination. So we have done steps 1 through 4. Now we have a completed polypeptide. We're on this step. We have a polypeptide, but there are some post-translational modifications. Those post-translational modifications can be folding or a chemical modification. So we have the number 1 for folding and the number 2 for chemical modifications. Folding is most likely going to happen in the endoplasmic reticulum. So I'm going to put a number 1 on top of endoplasmic reticulum. Chemical modification is most likely going to happen inside the Golgi apparatus. So I'm going to draw a number 2 there. So I know most folding happens in the endoplasmic reticulum as we are creating the polypeptide, and most chemical modification happens in the Golgi. So if I'm talking about primary, secondary, tertiary, and quaternary structures, I'm talking about folding. If I'm talking about ubiquination, methylation, phosphorylation, hydroxylation, these are chemical changes. So I'm going to draw a 2. If it happens automatically or with the help of a chaperonin or a chaperone protein, this is folding. If this happens in stages as vesicles travel through the organelle, well, the organelle we're talking about is the Golgi apparatus. So this is a chemical modification. So for number 5, all we had to do was label 1 or 2. 1, 2. Let's move on to the next step. In this next step, we're going to be going through the stages of DNA extraction. So we're going to follow these steps in detail. The first thing that we're going to do is fill a test tube about halfway full with 95% alcohol. Now 95% alcohol is much stronger than rubbing alcohol that you get at the pharmacy, which is normally at about 70%, 50 to 70. Then we are going to mix the extraction fluid, which includes 2 teaspoons of soap, just normal detergent. You can use dishwashing detergent. And then 1 teaspoon of salt. Then we're going to add distilled water. It doesn't matter how much distilled water, but we can add more later if we need it. We're going to pour all of this into a Ziploc bag. Then we're going to put a strawberry into the Ziploc bag and then smash it. The 4th step is to pour all of this mess and junk into a coffee filter, and that coffee filter will drain, catching all of this junk and only leaving this fluid behind. That fluid is important, that's what we want. Step number 5 is to add the alcohol that we prepared in step number 1. We're going to add the alcohol and then we will watch it separate, and we are going to collect the DNA on the end of a paper clip. So this is the process of DNA extraction that we will be using. Then we'll come back and answer these questions. So here I have all of the materials that we need. I have strawberries, ethanol, soap, salt, water, and then my paper clips and my coffee filter. And we're going to be working on this together. Let me make sure that this tripod stays still. Okay, the first thing that I need to do is make sure that I chill the ethanol, because the ethanol needs to be very, very cold. And so here I have some ice water, and I'm just gonna fill this with ethanol and chill that ethanol in ice water. The colder, the better. This is cold water. The next thing I need to do is add salt and dish soap to my Ziploc baggie. So I'm going to kind of prop open my Ziploc bag so that it's easier for me to work with. I need 2 teaspoons of soap. It doesn't have to be exact, but we want it to be enough. Now you might be wondering, why are we adding soap? Well, we learned at the very beginning in the first unit of class that detergent or soap is really great at breaking apart fat. So we're going to use this to break apart fat molecules. Think about which organelle has a lot of fat molecules in it. Next, we're going to be adding 1 teaspoon of table salt. Now, we also learned in the first unit that table salt has a positive and a negative ion. Those ions are going to be really important because the ions help to extract the proteins and neutralize the DNA. If you remember, DNA has kind of a negative charge to it. All right, so now we're going to add in our strawberry, and we're going to kind of remove, well, we got to add a little bit of water just to mix everything up. And again, the amount of water that we add can be changed later, and I'll explain why when we get to the filtration section. But right now we just want enough water. So I'm going to remove as much air as possible, and then do the fun part where I smash our lovely strawberry. Now, this is called mechanical separation. If the dish soap is breaking apart the fats and this is extracting the proteins and neutralizing the DNA, mechanically separating it is just going to make it easier for the salt and the dish soap to do their job. It also helps to break apart the cell wall. Remember that strawberries are a plant, so they have a cell wall, and I want to do a good job of smashing this strawberry. I actually think that I might want to add a second strawberry because it doesn't seem like there's very much gunk that I'm extracting. So I'm going to add one more strawberry just because this step is really fun, to be honest. There we go, that's what I wanted. I wanted it to turn nice and pink, and I also don't want to smash it so much that my bag breaks, but I do want a nice pink, consistent, frothy liquid. It should look kind of like a smoothie, and I'm just breaking apart all this cell wall, all this mechanical structures. All right, when I have a nice pink fluid, I'm going to set up my filter. So I'm going to come over here, and I'm going to place the filter inside this cup and kind of clip it to the sides so it's easier for me to pour. And I'm going to pour my smoothie into the filter. This step might take a good minute, depending on how chunky your solution is. But the goal is to get as much of this liquid as possible down through the filter. So I'm just kind of gonna agitate it just a little bit, making sure not to break my filter. And my goal is to get as much of that liquid separated as possible, separated from all of this cellulose, essentially. If I need to, this is the point where I might want to add some more water if I'm having a hard time separating it, but I do think that I have a good amount right now. Having this pink liquid at the bottom is the goal. Now, in the instructions it says that I can make a packet. That accomplishes the same thing as agitating it, so I'll do that now. I'm going to remove the clips and kind of fold my filter into a little packet. And you can use the clips to clip this packet closed, and then that actually does speed up the process as well. and then we can kind of squeeze out some of that water, some of that liquid. All right, I'm going to set this to the side, and I have a good amount of liquid at the bottom, which is what I wanted. Now, it doesn't look like very much now, but what I'm going to do is take my paper clip and unfold it so that I end up with a little hook. My fingers are a little slippery from the soap, but I want a nice little hook like this so that when I can extract the DNA. Now, believe it or not, there is DNA inside of this liquid. Okay, there is DNA in there, but I can't see it yet. It is dissolved. So what I'm going to do is I'm going to very slowly add the alcohol to the edge and slide it down the edge. I'm not going to stir, I'm just going to slide it down the edge. And it will create kind of a layer. In your cup. Notice how there's a clear layer on top of your pink layer. This is going to cause the DNA to separate, and I am going to leave it there for a minute or so without agitating or stirring it. And we might notice that we're going to start to see more and more separation and start to see some development of a white gunk. That white gunk or snotty looking substance is the DNA strand. And strawberries are really, really great because strawberries are something called polychromosome, where they don't just have 2 copies of their chromosomes like humans do. They have many, many copies. Let's see if we can extract a little bit of DNA here. So I'm gonna kind of fish it out. So that is DNA. I see a good strand right there at the bottom. Let me see if I can hook it up. There we go. This is the DNA, and this is actually what Rosalind Franklin did to extract DNA, and then she held up a strand. Imagine how labor intensive that was to do this over and over and over again until she got a single strand. And then she shone an X-ray across it to discover that X photo 51, or the double helix shape. So this, believe it or not, is DNA of a strawberry. And we can even get more of it if we kind of fish around for it. Now, as it warms up, it becomes less and less stable. That's why it's important for the alcohol to be very, very cold, because you can see the DNA in this solution. That's the DNA right there, but I can't pick it up because as it warms up to room temperature, it becomes less stable and I'm not as easily able to hook it onto the paper clip and pick it up. So the colder the better. In fact, one year we used frozen strawberries, and the frozen strawberries worked really well just because it added another element of cold. Now, all of these materials are household materials. So if you want to try DNA extraction at home, if your roommate or parents don't mind you making a little bit of a mess, all of this can be found at home. It's okay if you use rubbing alcohol. It's 70% ethanol instead of 95% ethanol, which will still work effectively. And you might want to try different fruit. I know bananas is a good option as well, not just strawberries. But let's go ahead and answer some of these questions that we have at the end. It says, to extract DNA from cells, what must you isolate it from? The other macromolecules. So we're going to separate it from lipids, mainly the cell membrane and the nuclear membrane. We're also going to separate it from protein and carbohydrate. Now, if I'm talking about carbohydrates of a plant, I'm usually talking about the cell wall. And those proteins pretty much make up everything else. So we have to separate it from the lipids, proteins, and carbohydrates so we can isolate the DNA. Now, let's talk about each step. One of the steps that we used was crushing the fruit. This is going to separate the fruit from the carbohydrate or the cell wall. The detergent is really good at breaking apart lipids. So we're going to use this to separate it from the plasma membrane or the nuclear membrane or lipids. The salt, the sodium chloride, the sodium has a positive charge and the chlorine has a negative charge. This will help to neutralize the DNA and proteins. The alcohol helps to separate out the DNA. Why does the alcohol have to be cold? Well, it needs to be cold for stability. And then the last question says, what is DNA used for once it's extracted? Well, we can use it to image, or we can use it to do DNA studies, such as gel electrophoresis. So we could use it for lots of different things. This is kind of an open-ended question. Well, thank you for joining me this week, and I hope to see you again next week for our last lab, Lab 3.4.